my name is Megan Gessel. I'm a graduate student here in the Bowers Group at UCSB. We use ion mobility mass spectrometry to study the size and shape and structure of different molecules in the gas phase. I study the amyloid beta protein, which is a very important protein that's implicated in Alzheimer's disease. This is an ion mobility mass spectrometer, and I'm going to show you how it works today. First, we load our samples into these really tiny little nano-electrospray tips. They're glass tips coated with gold, and we only need 5 microliters of sample. Once we have the tip, we put it in this little tip holder and introduce the sample at this end of the instrument. The sample is ionized by nano-electrospray ionization, and once the ions are formed, they enter into the instrument and are stored into an ion funnel. They pass through the ion funnel, and then they come into the drift cell. So this long white tube is our drift cell. It's filled with about 13 torr of helium gas, and the ions pass through the drift cell, and after the drift cell they reach the quadrupole mass filter. And here they are mass selected and they can be detected. We also have a time of flight on this instrument here, which we can also use to add additional mass selection in our experiments. So with this instrument we can do 2 types of experiments. We can do a mass spectrometry experiment or an ion mobility experiment. In mass spectrometry, we let the ions just pass directly through the ion funnel, be mass selected by the quadrupole and detected for your normal mass spectrum. To add ion mobility separations, this way we can actually separate mixtures by their sizes and shapes. To do this, we pulse the ions in all at the same time into the beginning of the drift tube, and they pass through under the influence of a weak electric field. Smaller, more compact ions in our sample mixture encounter fewer collisions with the helium gas, whereas larger, more extended ions encounter more collisions with the helium gas. And so we can actually separate these by their sizes as a function of how soon they arrive at the detector. Those smaller, faster moving ions reach the detector sooner than the larger, more extended ions. And this is how we can do our ion mobility experiment.